Okay, very good morning, all the participants and respected Rajni Madam Garu, Associate Professor, Department of Botany, Food and Nutrition, RBVRR Women's College Autonomous. Madam is going to explain yesterday about the general characteristic features and also the most important reproduction, the structure of heterocyst. Madam has clearly explained. Now, Madam is going to explain the remaining type studies in blue-green algae. Madam, please welcome and share your screen.
Yes, sir. Yeah, it's clear, sir.
Madam.
Yes, yes. What is the good morning to one and all? Good morning, students.
Good morning, ma'am.
Good morning. In the last yesterday class, I'll explain to start the cyanobacteria. You know what is the cyanobacteria? Three names are there. No, one is the MIXOPHYCEAE and blue-green algae and cyanobacteria. In that cyanobacteria, the pigmentation and reserve food material, what type of reproductions are there, and what is the economic importance of cyanobacteria. In yesterday class, we will discuss these are things. Now here in this cyanobacteria, type studies also there, NOSTOC, Anabina, Oscillatoria. In that type studies to explain in this today class. The first one is the NOSTOC. Here, the first type study name is called the NOSTOC, or in this one, first you can observe the NOSTOC diagram. See, this is a NOSTOC picture. Just beads-like structures are there. You can observe pulse or bead-like structures is there, no? In that beads or pulses-like structures are present in the NOSTOC. See here, you can observe this one. Okay, these are the NOSTOC structure or habitat. Coming to the, what is the introduction? The NOSTOC thalli are present in the form of a colony. You know, what is the colony? These are things are already have discussed about the thallus organization also. The thallus is a NOSTOC is a form a colony fruit. NOSTOC is the filamentous cyanobacteria or blue-green forms both terrestrial and aquatic habitat. What type of habitat is there? Terrestrial algae, aquatic algae, and the freshwater algae also. In this one, here the thallus, the type of thallus is the colony. The main is the habitat is the colony is there. And sometimes these colonies, single or a large colony, single filamentous colony are there, and large colonies also present. And where there's one habitat, main important to water, where the water loggings are there, ponds, ditches, dam soils and reservoirs. These are all found in this habitat. And few species are the intracellular cavities also, where the duct webs are present. In that duct webs, the intracellular cavities are there. In that cavities, some species you can identify the NOSTOC. And here, the NOSTOC is a mostly important for the characteristic is the colony. This colony also ball-like colony. What type of colony? Beads or ball-like colony is enveloped by one gelatinous sheath. 1 second. Yeah. See, this is a colony. This is not film, only single one. You can observe this is a colony. Each and every colony to made up of the one mucilaginous sheath is present. That sheath is called the gelatinous sheath. Balls are green in color. See, this one, these balls are green in color, sometimes bluish in color. Each and every colony contain thousands of straight or twisted filaments. See, you can't see here in the just ball-like structure, just one bunch-like structures is there, no? In this one, each and every colony, in this one, there are many colonies are there. Each and every colony contain a thousands of straight or twisted filaments or trichomes also present. Where the trichome, see, in this one, this is a filament. In that filament, in middle part to form to the trichomes. Each trichome surrounded a individual sheath and that is called the filament. That a trichome is contracted and consists many cell arranged in the beaded manner. Each and every cell, you can observe the NOSTOC cell, that is a cylindrical or spherical in shape. Filaments, there are present some large or spherical, cylindrical, colorless, empty. These are called heterocyst. Heterocyst, you can observe that's one. 1 second in picture is there. Heterocyst, see, this is a colony. This one is the colony, and here also colony is there. These are the thallus are present in the form of colony. All ball-like colony enveloped one gelatinous sheath. Each and every ball, greenish color, bluish color is present. Each and every colony contain thousands of straight or twisted filaments are present. Here, one colorless is there, no, in the picture you can observe, that is called the trichome. Each trichome is surrounded by a individual sheath. This is called the filament. And another one, in filament, there are present some large spherical cells. This one, large spherical or surrounding cells are there, colorless, empty cells. This is called the heterocyst. See, this one, you can observe in the picture. Here, these all rounded beads are there, no? In that rounded beads is the bluish color, greenish color is there. In between the rounded beads, you can observe this part, spherical shape or cylindrical shape is there. This cell you can observe, this is only colorless cell. This one is called the heterocyst. These heterocysts are generally intercalary, but in the end condition, they be terminal. Two polar nodules are present in each heterocyst. Some cells of the filament become enlarged and filled with the food material. These are thick-walled cells. That cell's name is called akinets. Both are there. Heterocysts are there and some are the akinets also present. See, this is the NOSTOC filament. One filament, Alex, no? This part is called the heterocyst. You can observe the different, this is very simple type study, NOSTOC, Anabina. In this one, the main important character, heterocyst and akinets. Heterocyst meaning spherical, cylinder shape, colorless, empty cell. Here not to the any filling thus not is present. Only empty cells are there. These empty cells are called heterocyst. Coming to the another one, heterocysts are generally intercalary, but the young condition, they may be terminal. This young condition, they may be terminal. The polar nodules present each, this is the polar nodules, present each heterocyst. Another one, after heterocyst, some cells of the filament become, you can observe these three. Where my arrow mark is there, in that arrow mark you can observe some cells of the filament become enlarged and filled with the food material. These, see difference in between the heterocyst and akinets. These akinets filled with the food material, very thick-walled cells is there. This is called the akinets. These akinets are generally present in chain model. See here, this is a chain model, present in a chain model in the akinets. And a single, you can observe, this is clear. What is the akinets, what is the colony? Yes. Coming to the next one, Each and every cell, you can observe the NOSTOC. Each cell surrounded by a cellulose cell wall. Each one surrounded by a cellulose cell wall. Protoplast shows a typical MIXOPHYCEAE structure. What is the meaning of typical MIXOPHYCEAE? That is inner colorless centroplasm, outer pigment chromoplasm is there. This is the important characteristic feature of NOSTOC or cyanobacteria importance. What is that one? The each and every cell to made up of the cell wall, cellulose, and inside the cell, the protoplast is present. This is the typical MIXOPHYCEAN structure is there. What is the meaning of MIXOPHYCEAE structure? That is, inner colorless centroplasm is present. Outer side pigmented chromoplasm is present. This is called the MIXOPHYCEAN structure, the cell. Not only this one, you can observe the CYANOPHYCEA total. Okay. Another one in the chromoplasm are pigments, proteinous, cyanophycinous granules, and cyanophycinous starch granules are present. While the centroplasm is present in that one, the centroplasm, the present of the nucleus also present. And this, where you can collect the NOSTOC, mostly NOSTOC can be found in soil, on moist rocks, at the bottom of lakes and springs. Springs meaning both fresh and saltwater springs. Rarely, very rare, very rarely you can observe in the marine habitat. What characters in this one? Here, NOSTOC, blue-green algae with arranged in bead-like chain that are grouped together in gelatinous mass. See this one. Okay. This one is together in a gelatinous mass. Size ranging from microscopic to walnut. You can't say directly. You can under electron microscope, you can observe this beaded-like structure. In the masses of these NOSTOC may be found on soil, floating aquatic water, this one place. Reproduction, fragmentation method. Here, special thick wall. You know what is a special thick wall? That one is the actinids, as the ability to withstand discretion of a long period of time. After 70 years of dry storage, the actinids of one species germinates into filament when moistured. See how many years to store the actinids are the stored food material. 70 years of dry storage. That's where like most blue-green algae NOSTOC contain two pigments, blue phycocyanin and red phycocyanin, erythrin, as well as chlorophyll. It has the ability to fix nitrogen in specialized cells. That cells is called heterocyst. See difference in between actinids and heterocyst. What the heterocyst function? To fix the nitrogen. It has ability to fix the nitrogen in specialized cell. That cell is called heterocyst. Some NOSTOC species, terrestrial species also present. In that terrestrial species has been used as the supplementary food source in Asian countries. That one in Asian countries, the food source also very important in this one, NOSTOC. In this, see, this is the single vegetative cell heterocyst. This one is a single vegetative cell and this is called the heterocyst. This is the cell and this is a protoplast. Each and every one I'll explain the before slide. No, see here this one is the total chromoplasm, heteroplasm inside the NOSTOC pictures. This one. And coming to the pigments also, I'll explain what type of pigments are there and what is the pigment system. These are things are completed. Now, this NOSTOC Reproduction, sexual reproduction is absent at all MIXOPHYCEAE member. Asexual reproduction, fragmentation, harmogonia, akinets, heterosis, endospores. This is the reproduction. Fragmentation, breaks up into the heterocyst and to adjusting cells into a number of fragments is called harmogonia. Each harmogonia to produce the new filament. Akinets also I'll explain in the before one where the akinets are there. What type of importance of akinets? See, this is the colorless part is called heterocyst. This color part is called the akinets. In the akinets, they store the food material. These are the reproductive akinets and heterocyst. Another very important in the NOSTOC in biotechnological importance also there. What is the biotechnological importance in this one? Here, NOSTOC has been as a dietary supplement for centuries. Several edible species is there. In the NOSTOC also several edible species. NOSTOC communis, NOSTOC spiridus, NOSTOC flagelliform have collected from various natural habitat from human use. Recently, the nutrition value of edible NOSTOC species has been characterized and number of pharmacological studies suggested that NOSTOC contain a bioactive compounds which with the anti-tumor, antioxidant, anti-inflammatory functions are present inside the NOSTOC. To meet the increased market demand, mass cultivation of NOSTOC has been attempted during the past decades also. This in the recently research of morphological development in the pharmaceutical values, growth, physiological of NOSTOC with an emphasis on the edible species has been successfully done in the NOSTOC. This NOSTOC also edible species has been successfully cultivated and pilot scale several challenges, several and technical challenges also present in this one. This is a colony, loss of colony, commercialization in the future of the NOSTOC. And coming to the NOSTOC, fragment reproduction already I'll explain. No, fragmentation, harmogonia, akinets. In this one, heterocyst and endospores also presents in this NOSTOC. Already I'll explain. No, this nitrogen fixing heterocyst are specialized mainly fixing of the nitrogen. Here filament these NOSTOC and anabina also to fix the nitrogen. This nitrogen is the form of dinitrogen method using the enzyme nitrogenase enzyme in order to the provide cell filament with the nitrogen synthesis, biosynthesis. Why this one? Yesterday sir told, no, this is the inactivated by the oxygen. So these heterocysts create the micro anaerobic environment condition. Anaerobic, why? Oxygen is not suitable in the fixation of the nitrogen. That's why this is a micro anaerobic condition of environment. Here, three additionals produce the additionals are there. That is a cell wall, including of glycolipids and form of a hydrophobic barrier of oxygen. That's why this is a nitrogenase enzyme involvement to fix the nitrogen fixation. And here, another important degrade of the photosystem 2. Two systems are there. Photosystem 1 and 2. In this one, degrade of the photosystem 2 with produced oxygen. The uprelated, that is a glycolytic enzymes are present. And another important to produce protein, the scavenger any remaining of the oxygen. That's why contain polar plug composed of the cyanophycin with the slow down to the cell-to-cell diffusion. This one is called the NOSTOC. I think so you can understood. Yes?
Yes, madam.
Yeah. The next one coming to the acylla